To round things off, we're now going to look at a cautionary tale of why experimental controls and pilot studies are a good idea. This is a story of Carl, a friend of mine and fellow PhD student. We were in the same lab doing our PhDs together at Oxford. Carl had a research question regarding how genetic history affects the repeatability of adaptive evolution. In our lab, we commonly use a technique known as experimental evolution, which is following populations of microbes, usually bacteria, as they evolve under certain laboratory conditions for many generations. And this is performed by repeatedly growing them over an extended period of time. In Carl's case, this was for 9 months. Because microbes can go in the freezer and then be revived, we can then compare evolved bacteria to their original ancestral state, and then ask questions about how things like bacterial growth rate and mutations that they accumulate change over the course of the evolutionary process. We do this at the end by using whole genome sequencing to identify which genes and which mutations have occurred. So Carl had an experimental design, which first involved a history phase of genetic drift, where the bacteria diverged away from their common ancestor. This was followed by an adaptive evolution phase, where the now diverged strains increased in fitness through acquiring beneficial mutations that improved their growth rate. Following this experiment, my role was to take the genomic sequences of all of these bacteria, see how they mutated, and investigate whether there was any similarity or differences that were found in the mutations that these diverged strains had acquired. At least this was the plan. What actually happened along the way was rather different. The initial history phase worked okay. Carl was able to generate a diverse collection of strains, and here represented by these 4 different colors. However, during the adaptive evolution phase, what happened was that one of the diverged strains, here the one shown in pink, contaminated the rest of the populations. This happens really quite frequently in these types of experiments because all it takes is a simple slip of the hand where a tiny amount of bacteria from one culture splashes and gets into the other tubes. Because each of these diverged strains had a different genetic marker, when it came to the genomic sequencing step, we identified that the cross-contamination had occurred. And this stopped us from addressing our scientific question, because we could no longer be certain whether any of the mutations we detected had arisen independently or were due to the contamination event. Furthermore, we lost some of the strains entirely. So this orange population here was completely replaced by the pink strains, and so we weren't able to even try to ask any questions from its genetic background. The consequences of this were rather moderate, I would say. As a result, we wasted about £5,000. This was directly incurred costs for the DNA sequencing. We also lost about 9 months of time, and this incorporated both his time doing the experiment and my time doing the analysis and figuring out that the cross-contamination event had occurred. He ultimately had to repeat this experiment, which again required more time and resources for the laboratory materials and the sequencing, and it delayed the submission of his thesis. Needless to say, he was really disappointed about this outcome, and ultimately it contributed to his deciding to not work in the area of microbiology anymore. He now does Parkinson's disease research, so you needn't worry too much. It's actually a positive outcome for him as a person. He's enjoying his life as a Parkinson's disease researcher. But at the time, it was rather soul-destroying. So how could we have avoided this outcome? Now, it's not possible to completely prevent these types of errors from occurring. You're dealing with billions of bacteria, and all it takes is one to get in the wrong place. But there are a number of steps that we could have taken during this experiment to make sure things were going according to plan. We could have included a negative control, and had a splash event occurred, we would have seen growth in the negative control tubes, indicating that maybe we need to be careful with the results. We could have done a pilot experiment first to check whether such splash events are likely to occur. During the experiment, we could have looked at the different phenotypic markers these strains possessed, as each of them actually had a different fluorescent protein that they expressed, so we could tell them apart at the end. Had we checked during the experiment, we would have known a lot sooner than when we sent the strains off for sequencing that something wrong had happened, that we should have gone back and restarted certain aspects. And finally, again, we could have checked these phenotypic markers before setting off the strains for sequencing to ensure that the correct phenotypes were found in the right tubes. To wrap up, in this session, we've identified that there are experimental designs that look ideal from a theoretical point of view, but they're not always the ideal experimental design to run under real-world conditions. Again, I'm not saying that you should forget everything we learned about ideal design, but don't let ideal design get in the way of conducting a successful experiment. Remember, we're always trying to minimize noise and extraneous error in our data, but sometimes the mistakes introduced by a complicated design can introduce another source of error. You have to consider the trade-off between ideal design and what's practically possible, and then use the appropriate statistical models that we've learned to compensate for this trade-off. Finally, we have experimental controls and pilot experiments that can help us reduce noise and uncertainty by giving us confidence in our data and our experimental designs. These will always be specific to your specific system, but can really help you to avoid costly errors and wasted time.